Hello, everybody, and welcome back to Creation Myths. We are here tonight for a live show to talk about a genuinely terrible video from Discovery Institute on junk DNA. Because this is a live show, we've got a live audience here. If you are watching this as I record on March 27th, 2024, thank you all for being here. Just let me know in the chat if the sound is okay. And when I pull up the video, we're going to be going over if there's any sound issues, just let me know. If you're watching this from the future, welcome. Thank you for watching. Don't worry about commenting about the sound or anything, because presumably it's fine. Today we have a really fun and thankfully pretty short thing that we're going to talk about. I want to give you just a few kind of channel kind of updates before that. So next week I have a pre-recorded video that's already ready to go. It's uploaded, it's scheduled. So we'll be right back here 1 week from now talking about a new creation trick. And if you are a channel member, you already have access to that video. So if you're one of those people, hope you enjoyed. Thank you for your support. And then the week after that, that would be April the 10th, would be that Wednesday, is I have a guest. It's Jordan, friend of the channel, and actual friend, Jordan, from Reason to Doubt. And we're going to be talking about the 2024 election, which I am really excited for that conversation. So those are the things we have coming up. I have a couple other things scheduled a little bit further out that I'll clue everybody in as we get closer to those. But that's what's coming up for the next couple of weeks. One recorded video, one live show in the near future. But for tonight, we are talking about junk DNA, and specifically this video that was recently put up by Discovery Institute. It's one of those animated videos from Long Story Short. So we are going to pull that up right here. Here's our video. And once I start playing this again, if you're here live, just let me know if the sound seems okay. So we're just going to go through this. It's pretty short. It's only like 7 or 8 minutes long. We're going to go through it. I'm going to pause periodically and point out where there are problems. And let me tell you, there are a lot of problems in this video. Some of them are very common for the junk DNA, capital D discourse, and some of them are less common. And there's at least one thing that's, I think kind of shady that goes on in here. So without further ado, I am going to hit play on this, and we're going to work through it. And yeah, here we go, everybody. This will be fun.
Okay. You know how you can tell a lot about a doctor with dead office plants? You can also tell a lot about a scientific theory by the predictions it makes. Here's the story. In 1972, this handsome fellow made a prediction that at least 90% of DNA appears to represent nonsense, and he coined the term junk DNA. Fast forwarding a bit, in 1980, Nature published papers by influential biologists arguing that evolution predicts our DNA should be full of junk. They predicted junk DNA would be everywhere and that it's folly to hunt for any function in these areas of DNA. Because
Okay, what do we make it? 30 seconds? 45 seconds? First big problem here. What is pictured is not junk. This right here, that's garbage. And there's a really important distinction between the concept of junk DNA and garbage. Because junk is stuff that doesn't cause a problem. Garbage is stuff that does, right? Like this, if you have this building up around your house, your apartment, wherever you happen to reside, that's going to cause problems. It's going to smell. It's going to attract pests like insects and rodents. It's going to be kind of an epicenter for disease transmission. Right? So this representation of junk as an overflowing dumpster, that is wrong, right? Junk DNA is the stuff I have in that drawer in my kitchen, those like weird little kitchen tools that I never use, right? For like coring an avocado. And that's the only thing that tool does, right? That's junk. That sits in that drawer and I never use it, but it doesn't hurt me to have it there. It doesn't like accumulate and attract flies and rats, right? I'm sitting here in my office in my basement. This is where my office is. Hi, I'm in my basement. And right over there on the other side of my basement, on the other side of the walls of my office, they're boxes. They're boxes that have things in them, like, you know, extension cords and surge protectors. Well, I have like a new surge protector sitting right over here on my desk. I don't need those 3 old ones over there. Those are just junk, and the likelihood that I ever use those for anything ever again is close to 0. But it doesn't hurt me to have that stuff over there. That is neutral with respect to how I live my life, right? But what is pictured here, this garbage, this is very harmful if this stuff was to accumulate, right? So like right away, we have a misunderstanding of the concept of junk DNA, and that does not bode well for the accuracy of this video going forward. And I also want to highlight a comment from the chat since we paused. This is true, right? The term junk DNA was not coined by Ono. It goes back to at least the 50s. What I always like to say is that it was first defined in the literature by Ono, because we can find it being used in the past, but at least to my knowledge, and Zach, correct me if I'm wrong here, but at least to my knowledge, we don't have a formal definition in the literature until Ono in the 70s. That's my understanding, is we find it kind of informally used before then, and then we have a formal definition at that point. So let us continue. And again, junk, garbage, 2 different things. This is a distinction that Dr. Dan Grauer makes, and it is a very important distinction when we're talking about this topic.
Because of natural selection, we should expect to find our bodies riddled with heaps of useless junk DNA. In the early 2000s, the Human Genome Project was finished, and we found out that only about 1 to 2% of our DNA builds the proteins we need to live. What in the world was the other 98% doing?
Oh, let's pause, because gold standard of science is making predictions and making accurate predictions. Anyone have an accurate prediction for what's going to come next? I think you all know what it's going to be, because you've heard this one before, right? Only 2% of the genome codes for proteins. We must have assumed that the other 98%.
It was immediately assumed that it was largely junk.
That is wrong. And I'm just going to say outright that that is a lie. It has never been the case that we looked at the genome and said, oh, this is the fraction that's protein coding, and the rest of it must be junk. That has never once been the case in the history of biology, genetics, evolution, any of this stuff. It just hasn't been. And the reason for that is that the concept of junk DNA as defined by Ono was defined in the literature, right, early 70s, prior to the discovery of non-protein coding regions of DNA that are functional. Okay, so we have tRNA genes, for example. I see in the chat somebody mentioned tRNA genes. Yes, right. We also had the elucidation of things like the lac operon in the 60s, involving things like promoters and operators, regulatory sequences that are non-protein coding but are clearly functional. Right? So it is simply wrong to say that at any point in this conversation, we assumed that the approximately 98% of the human genome that is non-protein coding was junk DNA. That never happened. And creationists say it happened all the time, but it simply never happens. I have a short video on that. It's linked down in the description if anybody is interested in just slightly more details there, but we pretty much just covered it.
Follows a long tradition of evolutionists preferring to view our bodies as poorly cobbled together assemblies of ill-fitted parts held together with not much more than spit and duct tape. You might feel that way the older you get, but that's not because of junk in your DNA. Look at your posture. Eat a vegetable every once in a while, for crying out loud. This textbook taught many biology students that over half of the genome is...
Oh, let me just, before we even get to this, I want to show you all something funny. That textbook, this textbook right here, the one that I use in my evolution class every summer, yeah, it's a great textbook. I really like this textbook. It has very nice, clear explanations. It has really nice figures. I really dig this textbook. Let's go back to the text, but I really dig this textbook.
composed of parasite-like segments of DNA and take up large amounts of space.
Let's just pause on that claim, because that is objectively true. Okay? It says over half the genome is composed of parasite-like segments of DNA, which is referring to transposable elements, mostly retrotransposons, which are approximately 40 to 45% of your genome. These are often called genomic parasites. They basically just copy and paste themselves, and they just take up space in your genome. There's another approximately 8% of your genome that's made up of endogenous retroviruses. These are the remnants of viruses that integrate themselves into your genome. These are undoubtedly parasite-like genetic elements. This statement right here is completely fair, right? So you take those 45% that's retrotransposons, another 8% or so that's endogenous retroviruses, and then there are other things. There's DNA transposons, there's other stuff that's also in these categories of, parasite-like genetic elements, right? So this is a perfectly fair claim. I don't know what the issue here is.
Famed evolutionary biologist Richard Dawkins said in 1976, "The true purpose of DNA is to survive, no more and no less. The simplest way to explain the surplus DNA is to suppose that it's a parasite." And in 2004,
that's confirmed prediction, by the way, because we hadn't sequenced it back then, right? 1976, we hadn't sequenced the DNA. Subsequently, we sequenced the human genome when we find that, yes, it are these retrotransposons, endogenous retroviruses, right? These essentially parasitic elements within your genome. Yes, so that is an accurate prediction from Richard Dawkins.
Creationists might spend some earnest time speculating on why the creator should bother to litter genomes with untranslated pseudogenes and junk tandem repeat DNA. In 2009, the greater part, 95% in the case of humans, of the genome might as well not be there for all the difference it makes. And Dawkins isn't alone. Prominent evolutionists, from the head of the Faraday Institute to the director of the National Institutes of Health and many tenured professors, all predicted...
Okay, we gotta pause right there. Predicted. These are not predictions, right? These papers and book, these are not predictions. These are descriptions of the evidence that we have regarding the functional fraction of the human genome, okay? There are not years shown here, but to give you an idea, this Grauer paper right here, this one at the bottom, the functional fraction within the human genome cannot exceed 15%. That is an, if I recall, 2017 paper. What this video doesn't mention is there was a correction and update to that paper published in 2020. No mention of that. This book by Larry Moran, Dr. Lawrence Moran, that book was published in 2023. This is not like, oh, we think this is what we are going to find in the genome. These papers in that book are descriptions of what we actually find in the genome. These are not predictions. Calling these things predictions is being dishonest. This is the shady thing I alluded to earlier. These things are not predictions. These works are descriptions of evidence. And what the evidence overwhelmingly shows is that a huge fraction of the human genome does not have a selected function for like human physiology. Okay, that's a big problem here. Like, there's no other way to kind of describe this. This little bit right here is a misrepresentation of what we're talking about. These are not predictions. I want to make that very clear for everybody.
Junk DNA. And remember, you can tell a lot about a theory from the predictions that it makes. Scientists that supported
Just going to pause for a second and highlight this comment. Zach, that's Dr. Zach Hancock, who is hanging out in the chat right now. He did a video with Dr. Moran. It's well worth watching. I agree. It's wonderful, and everyone should check that out. The channel is Talk Pop Gen. You should check out that interview with Dr. Moran. It's about his book. It's very informative. Highly recommended.
Intelligent design, though, had different predictions about junk DNA. Ever since the beginning days of the ID movement in the 1990s, ID proponents had predicted that the junk DNA would turn out to be largely functional. What would cause these scientists to go against the tide of decades of established scientific thought? Why does ID predict function for junk DNA? It's simple. Because intelligent agents make things with purpose and function. They have reasons for why they do things. Perhaps the earliest ID prediction of...
I do want to point out a contradiction here. Anytime you point out something that doesn't seem to work efficiently to an intelligent design creationist, and yes, I'm going to call them creationists because, like, that's what they are, or if you prefer, see design proponentsists. But anytime you point out to an idea that there is something like poorly designed, right? Like the human ankle, for example, the spine, it's not good. There's a lot of elements to the human body, right? The optic nerve blocking part of your retina. There's a lot of things that you can point out and say, well, that's not good design. And what we often hear in response to that is, well, how can you know the intentions of the designer? Maybe we just haven't figured it out yet. Well, okay, fair. Let's take that at face value. Then how can you know the intentions of the designer and say they would make something with, a high degree of functionality, right? Can't have it both ways. Either you can, make predictions about the intentions of the designer, or you can't. But you can't have it both ways. Discovery Institute. And it is also worth noting here that lots of biologists, hang on, let me find the comment here. It's going fast. I got to scroll back a little bit. Here we go. Thank you, Zach, the previously mentioned, at Talk Population Genetic, Talk PopGen on YouTube. Zach Hancock, a lot of biologists prior to intelligent design, which is like a 90s kind of thing, they argued against the concept of junk DNA. It's the adaptationist camp, if you want to kind of do a bit of a dive into that. That's who we're talking about here. So it wasn't just the ideas who were saying that, as other biologists, like real biologists, not creationists.
A function for junk DNA came in 1994 when pro-ID scientist Forrest Mims submitted a letter to Science warning against assuming that junk DNA was useless. Science would not publish the letter. In 1998, William Dembski predicted function for junk DNA, arguing that even the term junk DNA discouraged scientific inquiry, a label that cloaked scientific ignorance with a veneer of confidence.
As we'll see in just a few minutes, that's clearly wrong. Like, very obviously, clearly, that was not the case, right? That this like stopped people from looking into it for some reason. That's just wrong, and we'll see a very, very important example of that in just a few minutes.
He says, on an evolutionary view, we expect a lot of useless DNA. If, on the other hand, organisms are designed, we expect DNA, as much as possible, to exhibit function. Design encourages scientists to look for function where evolution discourages it. In 2002, biologist Richard Sternberg published a peer-reviewed paper in the Annals of the New York Academy of Sciences, stating the selfish DNA narrative and allied frameworks must join the other icons of neo-Darwinian evolutionary theory that, despite their variance with empirical evidence, nevertheless persist in the literature. In other words, the idea of junk DNA was like an idol in the scientific community. Empty, but nevertheless handed down due to tradition rather than evidence.
Okay, let's pause and talk about this for a minute. Tradition rather than evidence. This is just a big pile of stinking nonsense right here. Like, just straight up saying that for decades we said, oh, well, in the 70s we said there must be a lot of junk DNA, and then now we're just going to go along with that with no evidence for decades. That's just wrong. I mean, as we sequence genomes through, because we did the human genome late 90s into 2000s, but we had sequenced other genomes in the lead up to that, and we found that, like, as we did larger, more complicated genomes, we found more stuff that didn't seem to have a function. Subsequently, right, as we've gotten really good at doing genome sequencing and genetic kind of manipulation, we've been able to do things like delete millions of base pairs from the mouse genome. I forget the exact number off the top of my head. It's like 30 million nucleotides from the mouse genome with no problem. Like, experimentally, those mice, they're just doing their thing. They're totally fine. So, like, to say that it's tradition, not evidence, is completely wrong. One more thing I'll mention is call back to something we talked about earlier with, parasitic genetic elements. As we could sequence the human genome, we can see that, oh, these sequences match like these transposable elements. These sequences match retroviruses. right? So no, it's not at all. This is just like misrepresenting the history of this concept, is that it's not the case that we've maintained this concept of junk DNA out of tradition rather than evidence. It's that over the decades since the 1970s, since we've really been able to look into it in a formal way, we've just gathered more and more evidence that this concept is legitimate, right? This is just a misrepresentation, and there's no other way to put it. And I will say, actually, let's do the next part, and then I will.
2004, biologist Jonathan Wells argued the fact that junk DNA is not junk has emerged. Not because of evolutionary theory, but in spite of it. On the other hand, people asking research questions in an ID framework would presumably have been looking for the functions of non-coding regions of DNA all along, and we might know considerably more about them. In 2000.
Okay. That's an important bit of nonsense right there. Because what this is saying is, well, IDers know to look for function in the genome, but those stupid evolutionists don't. Like, they're not going to do it because it, violates their paradigm or whatever. But here's the thing. There has been a lot of effort to understand all the stuff that goes into the genome. And, like, people have been looking into it. Real biologists, not creationists, real biologists have been looking into it. As we sequenced the human genome, the original predictions were that we would find 100,000 protein coding genes. And as we sequenced the genome, we had to accept the evidence that that number was getting smaller and smaller and smaller over time, as we kept sequencing regions and finding fewer genes than we expected to find. And then, of course, there's the elephant in the room that we haven't talked about yet, but it's coming up soon, the ENCODE project. The ENCODE project, for all its problems, is real biologists, right? They're not ideas, they're not creationists. Those are real biologists. They're wrong in a lot of ways. They get way out over their skis, as we'll talk about. But like, the ENCODE project is made up of real, serious biologists who are not like ID proponents or anything. They're doing like actual work in biology, trying to find functions within the human genome. And that the existence of the ENCODE project just shows that what we just heard was wrong. It's very simple.
In 2011, Wells wrote The Myth of Junk DNA, citing hundreds of peer-reviewed papers finding function for junk DNA. In 2012, a game-changing paper was published in Nature from ENCODE, a consortium of hundreds of researchers from all around the world. It found that 80% of DNA shows evidence of functional biochemical activity.
Functional biochemical activity. That phrase is so misleading, because there is a difference, and many of you have heard this before, there is a difference between biochemical activity and function, right? Lots of things have activity that don't have function. Those transposable elements I talked about before, a whole lot of those do like a little bit of transcription. Doesn't do anything. The RNAs break down pretty quickly. They don't last. There's not a lot of them, but like, that's biochemical activity, but it certainly isn't function. Another example of this is histone binding. A big chunk of the human genome at any given time is condensed in this state called heterochromatin in the nuclei of your cells. Heterochromatin is DNA that's wrapped up around proteins called histones in a condensed state. And the reason it's in that state is because when it's doing that, it's biochemically inert. It's not doing anything. It's not being expressed, right? There's no other proteins that can get in there and activate that DNA. They can't bind to that DNA and do other stuff with it. And that matters because the DNA that is wrapped up in that dense heterochromatin state, it's not doing anything functionally. And for a large part of the human genome, there's giant chunks of it that are just persistently across your cells. They're just all the time in heterochromatin. That stuff is going to pop on ENCODE's filter because binding to histones is a biochemical activity. But it doesn't tell you that that DNA is functional. the specific nature of that activity, right? Binding to histones and then coiling up in this dense heterochromatin state, that's the opposite of function. That tells you that that DNA is non-functional. But here we have this phrase, 80% functional activity. It just throws all of these biochemical activities together into one bin. I mentioned earlier Dr. Dan Grauer, who's done a lot of work kind of showing where ENCODE goes wrong. And he makes, I think, a really interesting point that's a little bit cheeky, I think he makes it, but it's also a serious point, that if the standard that you're using to assign function is biochemical activity, then the whole genome is functional because the entire genome is replicated. That's a biochemical activity that requires specific binding to enzymes and other things going on, right? That's a biochemical activity. Therefore, the whole genome is functional. And here's there you go, Zach, making the exact same point. On the same page there. Yes, exactly. Right. If this is the standard we're using, it's curious that we omit replication from that consideration. It should just be all functional by that standard. So this phrase, functional activity, is extraordinarily misleading.
Well, so that means 20% is still junk, right? Well, no. Lead ENCODE researcher Ewan Burney noted that they only studied a portion of human cells, and with continued research, it's likely that 80% will go to 100%. The implications were...
I want to note that was from like a press release, not like any kind of peer-reviewed paper. And there's no, that is just a tremendously ignorant statement to say that 80%, if you take the 80% at face value, which you shouldn't, it's going to go to 100%. That is just absurd to the highest degree and so wrong.
Were immediately clear. Science magazine ran a headline stating, ENCODE writes eulogy for junk DNA, and stated that the ENCODE papers sound the death knell for the idea that our DNA is mostly littered with useless bases. Apart from a zealous faction of hardline evolutionists, the consensus of biologists now recognizes that junk DNA isn't junk, but has important functions. In other words, junk DNA was an evolutionary myth.
So that whole bit about consensus, that is so wrong. Most biologists accept the idea that some fraction of the genome is non-functional, right? Is junk DNA. That is the mainstream position, is that junk DNA is a real thing. That 80% going to become 100%. Not even ENCODE believes that anymore, as we'll see. And in fact, we're talking about the 2012 ENCODE paper here. And those of you who are familiar with ENCODE know that that was not the only paper they published. So in the spirit of making predictions, because, science, anybody want to predict what paper from ENCODE they will not be bringing up in this video? A subsequent paper that maybe showed that 80% goes to 100%, or maybe it super doesn't show that, right? Do we want to make a prediction? Somebody already did. Andrew knows the deal here, right? Are they going to bring up the follow-up from 2014 from ENCODE? Are they going to bring up the next paper? No, but we'll talk about it after the video because it's worth looking at. Because ENCODE doesn't even believe the things that ENCODE is saying about junk DNA at this point. The 2014 paper walks a lot of this stuff back. We'll talk about it. We'll get there.
Recent papers confirm this. This 2021 paper declares the days of junk DNA are over. This article in Nature 2021 finds over 130,000 specific functions identified for genomic elements.
Something I want to point out there. Actually, let me let them finish the sentence first.
Previously called junk DNA.
Okay. 130,000 functions for genomic elements previously called junk DNA. Here's the thing. There's a lot of DNA in our genome that falls in this category of junk DNA, even before ENCODE. Let's say you take ENCODE at face value. Prior to that, junk DNA, most of the genome. Now, you can look at all of those elements post-ENCODE. You can look at transposons. You can look at endogenous retroviruses with their long terminal repeats. You could look at this or that element in the DNA. And you can find examples of those elements that have function, right? You can find examples of those elements with function. You can find examples of endogenous retroviruses that have specific functions. We've co-opted them to do a specific thing. You can find examples of transposons that have a specific function. But most members of those different classes of genetic elements don't have those functions that a minuscule number of cases have been documented to have. And the reason we know this is that most of these genetic elements are actually degraded to the point where they can't do the functions that are assigned to some members of that class of elements. So for example, you can have endogenous retroviruses where the structure, little background, the structure of an endogenous retrovirus, it's a virus that integrates itself into the genome. At either end of these elements, gotta get by, there you go, you've got these things called long terminal repeats, LTRs. And then in between, you have three genes, GAG, POL, and EMV. And there are cases where you can find an endogenous retrovirus where one of two things is the case. Either those LTRs are regulating human gene expression in our genome, or one of those genes has been co-opted to do a thing, right? We now use it for us. But most of those things, most endogenous retroviruses are so degraded that they can't do those things. Those genes are mutated, and in most cases, they're actually missing. Those LTRs, they're mutated to where they can't regulate gene expression anymore. In most cases, it's just what's a thing called a solo LTR, where the whole rest of the endogenous retrovirus has been lost. And it's just one little LTR, and that's often mutated to the point where it can't do its original function, right? It can't do any regulatory thing. So you can point to these cases, and this 130,000 is an exaggeration, but you can certainly point to examples of endogenous retroviruses or retrotransposons that have functions. But the vast majority of members of those classes of genetic elements in the human genome are actually mutated. They're degraded to the point where they are unable to do the functions that people say, oh, this is what transposons do. Most of them physically can't do that thing. The sequence is not there. Another thing that they propose for these is an immune function where they say, oh, well, the virus-like sequence, it matches the virus and then it inhibits the viral gene expression. In order to do that, it needs to be under purifying selection, meaning selection to keep the sequence the same, because that function only works if the sequence in your genome that looks like a virus matches the actual viral sequence. If they're too different from each other, the interruption function doesn't work. Well, those sequences are not under purifying selection in our DNA. And again, most of them are degraded to the point where they wouldn't work anyway. So clearly, that's not something that's happening here. And it's silly to say that based on the actual sequences that are present in the human genome. So that's the problem that we're running into here now. They're saying, oh, look at all these functions that we've documented. Yeah, if you take your junk DNA, right? We've documented a function in this much of it, and we've also shown that the rest of it can't do those functions based on its sequences, right? There's other classes of things in the genome that fall into this as well, like pseudogenes. Thank you for pointing that out, Andrew. Love my audience participation. Thank you, Andrew. Yes, pseudogenes are in the same category, right? They can only do these functions as regulatory elements if the sequence of the pseudogene is selected to match the similar real gene. But we see that those sequences are not being maintained via purifying selection, which means they're not doing the function, right? That's it.
This article says the genome is far from what was once called islands of genes among intergenic deserts.
That's actually still accurate, even if you assume everything's functional, because intergenic just means between genes. And nobody's proposing that there's all these protein coding genes in there. So that's just silly. I don't know if maybe the narrator here, it's long story short is the narrator. I don't know the person's real name, but it's long story short, does these videos for Discovery Institute. Maybe he doesn't understand what intergenic means, but that's still true, even if ENCODE is right.
James Shapiro, University of Chicago. The concept of abundant, selfish, or junk DNA in complex genomes is mistaken. This 2023 paper in Bioessays declared a paradigm shift away from the concept of junk DNA. DNA that was previously thought to be junk turns out to have many functions, including
forming telomeres, centromeres.
Okay, before we get to this silly part, I do want to point out that most of those papers were before the book by Lawrence Moran that we saw earlier. That book, remember, was a prediction, but that book was published in 2023, right? Those papers, those 2020, 2021, there was one from 2023. Those papers were actually before the supposed prediction, which again, just shows you how those things that the narrator showed are predictions. They're not actually predictions. They're actually explaining the evidence, right? That book took all this stuff that we're hearing about into account and still demonstrated that approximately 90% of the genome is non-functional. Now, this part is kind of funny, and you'll see why in a second. Let's play through this here. This is all the functions that we found for junk DNA.
And higher order nuclear structures, binding cohesin to chromosomes, nucleation centers for DNA methylation, chromatin condensation, cellular stress responses, cell differentiation, DNA repair, controlling development and forming body plans, controlling the cell cycle, forming fear-related memories and phobia, immune-related functions like fighting virus infections, metabolism-related functions.
in the biggest.
So do you see what he did there? He made his voice higher in pitch to make it look like he was saying it really fast, but he didn't have to because it just wasn't that many things and it wasn't scrolling that fast. But just to like make it seem like there are more of them, he just made like the end of a like the end of a car commercial, but didn't have to do that. The other thing, I just want to go back just a second and point out how vague some of these are. Right, metabolism related functions. Can we be a little more specific, please? Forming fear related memories and phobias. Really? Because now we're getting all into like EvoSec. Like, citation needed, please. I'll believe it when I see it. Let's keep going.
Like fighting virus infections, metabolism-related functions.
And the biggest and most important function for junk DNA is that it regulates gene expression. To understand the importance of what this means, consider building a house. Proteins would be like the wood, the bricks, the nails, the concrete. But if you want a house, it's not enough to just have those things laying in a pile. You need to know how to assemble it all. In other words, you need a blueprint. Instructions to tell you how to build the house are just as vital as the physical materials themselves. This is what the non-coding regions of DNA, what we used to call junk DNA, does in your body. It contains the blueprints and tells your body when and where to produce different types of proteins, how much to produce, where to put them, and when to stop making them. In other words, biologists for a long time, and many still to this day, discount some of the most important genetic information in your entire body. The story of junk DNA.
Okay, so let's just pause on that for a second, because this is again an example of creationists taking small examples, right, of a type of DNA sequence that does a thing, and then extrapolating it to the entirety of that class of genomic elements, right? It is true that there are endogenous retroviruses that are regulatory. It is true that there are retrotransposons that are regulatory. It is also true that most of the members of those classes of genetic elements are so degraded, they are unable to do that function. They simply lack the sequences that are required to be able to do it. So it's just wrong. I'll also point out that it is 2024, and just spoiler, we're not going to get any more of like ENCODE in here, including like follow-up things like the 4D genome and stuff. And that's the fallback position that a lot of better informed creationists have actually taken, is that, well, it's not about regulating this or that, it's about the spatial arrangement of DNA, and you need these specific kind of geometric kind of secondary structures within the DNA. And even though those sequences are not functional at the sequence level, physically, those pieces of DNA are still important for, like, regulating where DNA is located in relation to other DNA in the nucleus of cells. That's actually something really interesting and does, I think, raise interesting questions about the definition of functional. I still think you could point to, like, again, small little bits that might, oh, this one associates with that one, so they're close together. That still leaves out most of the genome. So it still doesn't do anything to undermine the concept of junk DNA or its validity. But it's at least a newer and an interesting argument that isn't just straight up misinformation like what we've been hearing. And this is the time where I get to once again say to creationists, please do better. Like, try a little bit instead of just kind of using the laziest, most predictable arguments possible. Please.
Shows that scientists who support intelligent design not only made a prediction that turned out to be true and helped progress science forward, but also evolutionary theory hindered science by discouraging inquiry for years in this very important part of our genome.
Again, that's just simply not true. We were looking for function through the entire time we were sequencing the human genome, and then ENCODE did its thing. They were not creationists, right? There was nobody saying, no, don't look for function. People were looking for function.
After ENCODE, most biologists today recognize that junk DNA is highly functional.
That is wrong. Most biologists today recognize that there is a large portion of the human genome that is not functional. Like, that is the consensus position in the field of biology. There is a minority that says the vast majority of the genome is functional, but they are a minority.
So how did evolutionists react? There were some intellectual objections to the evidence, but mostly they got angry. They tried to rewrite history, or they tried to deny the data to salvage their beloved theory. But more on that in the next video. Thanks for watching. If you like this kind of...
Okay. Well, that was fun, wasn't it? We are not done yet, because one thing that we didn't hear about in that video, inexplicably, except, we know exactly why we didn't hear about it, but one thing we didn't hear about in that video is the 2014 ENCODE paper. So there was the 2012 one that was the big splashy one. And then 2 years later, the same team, ENCODE, they published another paper. And I'm going to pull that up now, and I'm going to show you. I'm going to show you what's going on here. So I'm just going to share this item real fast here. As soon as I can find it over here. I have that paper open somewhere. Stand by, everybody. I got to find that paper. There it is. Okay, here we go. This is what we're talking about. So let's talk about this paper right here. So we're going to pull this up and put that on screen. So let's talk about this paper right here, Defining Functional DNA Elements in the Human Genome, Kellis et al. 2014. This is basically the follow-up to that big 2012 paper. And this is really, really important for a few reasons. So the first thing I'm going to show you here is figure number 1, because I really like figure 1. One thing that the ENCODE team does really well in this paper is that they're really clear about what would and would not constitute function, right? These are the things that would show something is functional, and these are the things that would indicate something is probably not functional. And they're pretty clear about that throughout this paper, weighing the different types of evidence and how we kind of bounce those off of each other. So in this figure 1 right here, what we can see is we're looking at the whole genome is the circle. And then the blue circles are levels of activity as documented by ENCODE. So you've got the darkest one is a lot of activity, then in the middle, and then very low activity. And you can see a lot of the genome meets that standard for low activity. And then in green, we have genetic evidence. So that's like if you do a knockout experiment, is there a phenotype associated with that? And then in blue right here, or in purple rather, we have our teeny little fraction that's protein coding. And in red, that's going to be evolutionary conservation. So sequences that are preserved across, in this example, all of mammals. That's what we're looking at here. So I think this is a really nice way to look at it, because if you have something, for example, that has a low level of biochemical activity, and it's not preserved, it's not under purifying selection, it's not similar across all these different genomes, then you can be pretty sure that that thing is not functional. If you have something that, when you knock it out, it generates a phenotype, if it has a high level of activity and it's preserved, right, across all of mammals, for example, then that's like pretty darn robust evidence that that thing is going to be functional. And I think they do a really good job in this paper of weighing those different kind of types of evidence and levels of evidence, which I think is why creationists hate this paper, because it basically walks back the very hyperbolic findings from the previous ENCODE paper, the 2012 paper, right? Because the 2012 paper was just like 80% potentially functional activity. So that's this whole light blue circle, right? That's what the 2012 paper is doing. This paper is much more circumspect and much kind of more reasonable in terms of inferring actual function and distinguishing function from activity. So what I want to show you all is down here in figure 2, because this is really key. And we're just going to focus, there's a lot going on here. Forget everything from here over, okay? So the DNA ACE bar right here to the right, you can just ignore that. I just want to talk about these 3 leftmost bars, the green and yellow ones, because those are the ones with RNA. So this is looking at the kind of the levels of transcription, right, for different parts of the genome. So what we have here across the y-axis is percent of genome. And then we have RNA that has a poly-A tail, that doesn't have a poly-A tail, and then all RNA. Now, don't worry about what that means. If you're not familiar with that, it doesn't matter. For what we're talking about today, it totally doesn't matter. What I want to show you is how the level of activity that they document here is actually indicative of mostly low-level spurious transcription that is indicative of non-functional sequences rather than functional sequences. Because one of the things we hear all the time, especially from creationists, right? So what we do here is creationism, right? Is that, oh, well, you know, 75, 80% of the genome is transcribed. That's a waste. Why would they allow that to happen if it wasn't functional? That's a waste of energy, yada, yada, yada. Well, let's look at what the actual nature of that transcription, okay? So what we see here is this measure of FPKM. Now, that is a very specific metric. You don't need to know what it means for this. It's read fragments per kilobase of exon per million reads. Don't worry about it. For what we're talking about today, that doesn't matter. What matters is this phrase a little bit further down here, is that note that FPKMs are not directly comparable among different subcellular fractions, as they reflect relative abundance within a fraction rather than an average absolute transcript copy number per cell. Depending on the total amount of RNA in a cell, one transcript copy per cell corresponds to between 0.5 and 5 FPKM in poly-A plus and whole cell samples according to current estimates. Okay, so that was a lot. Let's break that down. Let's look at these graphs. What that's telling us is that low-level transcription, which we're going to define as one transcript or less per cell, which we can just say, like, if there is one copy of RNA of a specific sequence in a cell, that's not having a physiological effect. Like, if there's one or fewer per cell, that is spurious transcription. Just straight up, it's not having a physiological effect. So let's look at this now. We've got greater than 0, 0.1, 0.5 is the third bar, and then 1, and then 5. The other bound they put there is kind of this kind of bright, like rainforest green right here. So what we can do, if we look at these fractions, let's just look at all the RNA. Let's just look at this third bar here, okay? Greater than 0, 0.1, 0.5. That gets you to about 30% of the transcripts of the genome, I should say. 30% of the genome is being transcribed above a level that might have a biochemical relevance to that cell, right? The rest of this up here from the 30 to the 75%, so that 35%, completely irrelevant to the physiology of that cell. Just straight up, those transcripts aren't doing anything. And that's the conservative estimate. Depending on the cell type, it could be as much as this 5 is indicative of one transcript per cell. That's all the way down here. That's this bright green bar right here. If you cut off right there, now you're only looking at 5% of the genome making transcripts at a level that might be physiologically relevant, right? So you're looking not at 75% of the genome has some functional transcription going on. No, it's at best 30% of the genome, and at worst 5% of the genome. And the rest of that transcription, just based on the amount of the transcript that's present, is going to be spurious, because a single transcript per cell is accomplishing nothing physiologically. But creationists don't want to talk about that one, right? They just want to say, oh, 80% of the genome is transcribed and leave it at that. No, you have to look at the level of transcription. And if you're not looking at the level of transcription, then you're missing most of the story. So there's a couple other things I want to point out down here, because one of the things we hear from creationists is about how, well, transcription is costly energetically, which is true. So any transcription is indicative of function. Well, no, because look at this line right down here, starting with a priori. A priori, we should not expect the transcriptome to consist exclusively of functional RNAs. Zero tolerance for errant transcripts would come at high cost in the proofreading machinery needed to perfectly gate RNA polymerase and splicing activities, or to instantly eliminate spurious transcripts. So what that ENCODE is refuting the creationist argument here in the 2014 paper that everyone pretends doesn't exist. But anytime any of you encounter in the wild creationists bringing up ENCODE and they say, oh, 80% of the genome is transcribed, therefore it's functional, energetically demanding, they would stop it if it wasn't functional. No, you can encode at them to show why that's wrong. And it's from this 2014 paper right here. They go on to say that basically, I'm just going to read a little bit more of this because I think it's actually really good to hear this from ENCODE, right? In general, sequences encoding RNAs transcribed by noisy transcriptional machinery are expected to be less constrained, which is consistent with data shown here for very low abundance RNA. Okay? And that ends right, that's the sentence I just wanted to finish right there. Okay, so like, this is not like me, some random person saying, hey, this is like ENCODE is wrong. This is ENCODE saying, we got a little out over our skis in that 2012 paper. And here we are in the 2014 paper, kind of a little bit of course correction here. But of course, that didn't come up in the video. Creationists never bring this up because this paper shows basically that, yeah, even if you look at these biochemical activities that are documented, they're actually by ENCODE's own standards, right, that they describe. They're likely to be spurious rather than selected, right? They're just happenstance and the remnants of these mobile genetic elements. Okay, so that's what I wanted to show you all here from that 2014 paper. The idea that creationists, they go through all this rigmarole with junk DNA, and at the end of the day, they always cite ENCODE, but they always leave out the 2014 paper. It's like time stopped in 2012. And that other paper just doesn't exist. But if you go to that other paper, the data produced by the ENCODE project and their interpretation of it refute the creationist arguments that they base on the 2012 paper. So I want to invite everybody who's watching to do that in the future when you encounter those claims in the wild. So this has been my longer than expected review of a really terrible video from Long Story Short and Discovery Institute on the topic of junk DNA. It was full of incorrect statements and I think outright misinformation. Thank you all for being here. Remember, we got another video lined up for next week. Members already have access to it. I've got a couple of live shows coming up in April. That'll be a lot of fun, so keep your eye out for those, plus some other stuff I have in the pipeline that I'll tell you about a little bit down the road. So that's where we're going to end it, everybody. Thank you all for being here, spending this time with me. I hope you enjoyed it. I hope you found it informative. Have a great night.